Hi, I'm Ayumi Yamada from RIKEN in Japan. I'm so happy to join this meeting today. The title of today's talk is Reversibility of Neurological Phenotypes in Kleefstra Syndrome Model Mice by Postnatal GLP Supply. I will talk about our research published in iScience last June and want to introduce my unpublished current work. Probably I do not need any introduction for Kleefstra syndrome. So our group identified the G9A and GLP complex, which encoded by the EHMT2 and the EHMT1 genes, and this complex mediates the methylation on histone H3 lysine 9 residue, and this methylation regulates the transcription. After Dr. Kleefstra group reported the EHMT1 gene is responsible for Kleefstra syndrome, we decided to study this disease too. So now we are analyzing the pathological mechanism and the possibility for fundamental treatment of this disease. So other group reported the EHMT1 heterozygous knockout mice and the brain-specific knockout mice showed the behavioral phenotypes. So we are also using our own EHMT1 knockout mice as a model mice for Kleefstra syndrome. First, we examined whether the expression of GLP protein can rescue the behavioral phenotypes in EHMT1 knockout mice. To think about the future clinical trial, we induce the GLP expression after birth, and this experiment allows us to examine whether epigenetic information can be altered in terminally differentiated neural cells by postnatal GLP supply. And furthermore, we want to elucidate the molecular mechanism of this disease. So the EHMT1 gene is essential gene for all of the cells in our body, but why deletion of EHMT1 gene causes the neurological phenotype so strongly? Today, I want to talk about this 1 and 3 mainly because many of the patients' families are joining this meeting today. So these are the genomic structure for EHMT1 knockout mice and inducible EHMT1 transgenic to express GLP protein. For knockout mice, we deleted the set domain, which is responsible for the methyltransferase activity. And for EHMT1 transgenic mice, we cloned the EHMT1 cDNA followed by the CAG promoter in Lothar 26 locus and put the stop cassette in between the LOXP site at upstream of EHMT1 cDNA. By combination with the Cure recombinase transgenic mice, we can control the GLP expression by locus and the timing. And we use these 3 Cure lines, and 2 of them are Cure ER lines. This line can induce the GLP by drug injection. So we can analyze when and where we need to express GLP to treat EHMT1 mice phenotypes. So this is a movie of one of the behavioral tests called the open field test. We put mice in the box and how see they behave in the new environment. This test can examine voluntary activity and anxiety. So wild-type mice is curious to the new place, so it immediately starts moving around. In contrast, the EHMT1 knockout mice just keep sitting at the corner and don't move like this. So as you see in the previous slide, the total distance traveled in 20 minutes test was shorter in EHMT1 knockout mice compared with the wild type. And this rearing number shows how much the mice are curious to the new environment. Here, the number of knockout mice is less than wild type. When GLP was expressed in whole brain from embryonic stage, both results are rescued by GLP expression shown in green. The rearing number was small in knockout mice, so anxiety could be high in EHMT1 knockout mice. So we examined anxiety using light and dark test. So the EHMT1 mice showed anxiety, so they are not going into the light area, so the time in light is smaller than wild type. And this phenotype is also recovered by GLP expression. So from this result, if GLP was expressed in whole body from whole brain from embryonic stage, we can rescue the behavioral phenotype in EHMT1 knockout mice. And this result also suggested the reduction of GLP in brain induced this phenotype. Next, we examine whether GLP-D expression after birth can rescue the phenotype. We use Kaggle system to express GLP in all cells in whole body. So GLP was induced by tamoxifen drug administration at juvenile 3 to 4 weeks old. After that, we waited 4 weeks and analyzed the behavior at 8 weeks old. And after that, waited another 2 months and we did the second round test at 16 weeks old. First, I checked the GLP expression in the brain by Western blot. You can see the increased amount of GLP protein in transgenic mice line. And we examined histone H3K9 methylation level by immunohistochemistry using the neuron marker NeuN and the astrocyte marker GFAP. Both in neuron and the astrocyte, K9 methylation level was increased by GLP expression. Using these mice, we performed the open field test and the light and dark test. At 8 weeks old, we could not see any recovery by GLP expression in open field tests. But another 2 months later, in 16 weeks old, both total distance and the rearing number were increased and wild type in blue and knockout mice with GLP expression in green showed no significant difference statistically. In light and dark test, the time in light was also elevated at 17 weeks old, but we cannot see such effect at 9 weeks old. So this is a summary for this experiment. So the expression in whole brain from embryonic stage, we can recover the behaviors. And the expression in whole body from juvenile stage, it took time, but it can improve the behavioral phenotypes. So this is the experiment using knockout mice, but it suggested the possibility for cure of Kleefstra syndrome. Next, we want to know which cells are required to rescue phenotype. We tried the expression of GLP in neural cell of frontal brain. Like GLP induction in whole body, we induce GLP expression at 4 weeks old and examine the behaviors at 8 and 16 weeks old. But as you see in graph, we could not see any improvement even at 16 weeks old. So here's a summary. The GLPD expression in frontal neuron are not enough to rescue the behavioral phenotypes in EHMT1 knockout mice. So GLP supply in non-neural cells would be required. But we wondered whether no effect at all in GLP supply in neural cells. So we analyzed the histological phenotypes in EHMT1 knockout mice. Well, not only the behavioral phenotypes, EHMT1 mice showed the histological phenotypes in brain. It's been reported the spine numbers in EHMT1 knockout mice is less than wild type, and our mice also showed a decreased spine number. In addition, we measured the spine head size. The head size in knockout mice is smaller than wild type. This indicates the knockout mice spines are immature. Why spines are less in knockout mice? We analyzed the microglia in the cortical region and detected the number of microglia is significantly increased in EHMT1 knockout mice. And these increased microglia were active home. So it suggests that active microglia prune the spines from the dendrite in knockout mice. We saw the activated microglia in EHMT1 knockout mice, so we speculate neuroinflammation was induced in knockout mice brain. So we examined the expression level of inflammatory-related genes. We detected the RNA level of caspase-1 and the downstream factor IL-1 beta were elevated in knockout mice cortical area. And from single nuclear RNA-seq analysis revealed that caspase-1 is increased in excitatory neuron and the microglia in knockout mice. We examined these histological phenotypes were recovered by neuron-specific GLPD expression, and we can see spine number and the microglia number and caspase-1 expression were recovered by GLP expression in neuron. But spine size is still immature. So we are thinking this could be the reason why neuron-specific GLP expression cannot improve the behaviors. If case phenotypes were caused by neuroinflammation, these phenotypes could be reversed by inhibition of inflammation. So we prepared the EHMT1 and IL-1 beta double knockout mice and performed behavioral tests. Contrary to assumption, you see the result in purple. Knocking out of IL-1 beta cannot rescue KS behaviors. We cannot rescue the behaviors, but same as neuron-specific GLP expression, IL-1 beta can recover the microglia number and the spine number, but still spines are small and immature. So this result indicated deletion of IL-1 beta pathway can reverse the histological phenotypes, but it's not enough to rescue the behaviors. This is our current model of EHMT1 knockout mice brain. Because of the decreased amount of GLP, the histone H3K9 methylation was reduced, and the inflammation-related genes are upregulated. This induced the microglia activation and followed by the excess spine pruning. These inflammation and other factors lead to the defect of neural network and KS mice showed behavioral phenotypes at the end. The data I showed today is the result using model mice, so we want to know whether neuroinflammation occurs in human patients. And we know that inhibition of inflammation is not enough to rescue. We need to elucidate other pathways involving KS phenotypes. And we know we need to stimulate the reconstitution of neural network to rescue the phenotypes. So now we are trying the environmental enrichment. This environmental enrichment is reported to induce the reconstruction of neural network and the neurogenesis. So now I will show the very preliminary data that I'm working on right now. So we choose the voluntary wheel running as an environmental enrichment. From 3 weeks old, we allow mice to play with running wheel for habituation. And then from 4 weeks old, we put wheel in each cage and allow mice to run freely. After 4 weeks, at 8 weeks old, we analyze the behavioral test. This is still preliminary data, but as you see this graph, the mice with wheel group, there is no difference from total distance between wild type and the EHMT1 knockout mice, and the anxiety phenotype in EHMT1 mice was also recovered by wheel running. Now we are analyzing what was changed by wheel running and how long it can keep this effect. So today's my talk's take-home messages is our data suggests that case phenotypes in mice can improve even after birth. So there is a possibility to treat brain dysfunction in KS if same finding also exists in KS patients. So these are the collaborators to help this project. So that's all. Thank you so much.